Thank you for having me here. And as Xin said, that I just arrived a couple of hours ago, which means that if you have tough question, right? So you can we can discuss this offline after I catch up on some of the sleep. It's my pleasure to speak at this function, and I have been doing data mining and bioinformatics for quite a few years, and today I appreciate the opportunity to share with you some of our recent research. So for this audience, I guess we don't need to emphasize this, right? Because this is a KDD audience. This is the annual event. And all I want to mention is that we all know and appreciate that the size of the data that we acquired, right, has grown more than exponentially and have way past actually the speed that the hardware improves. As a result, the gap becomes larger and larger, which means that we can't really just sit there and wait for the computer to become faster and better to solve our problem, right? We have to actively engage ourselves to develop better algorithms and models to solve the problem, okay? So in our lab, we have been, I think it's almost, it's more than 15 years, close to 17 years that we have been starting to do actually the data mining in scientific field. And one of the large areas that I have been looking into a lot would be the biomedical science. And I just took some of the pictures randomly from papers from myself and colleagues, and then to basically illustrate what other problem that we have been studying. Right on the far left side would be the traditional actually computational biology, that we look at data, actually collect data from all level, all the way from a molecular level to the like tissue level, even population level. But lately that people appreciate the data have been collected from, for example, from hospitals and large cohort studies, and also those clinical data and data from maybe pharmaceutical companies, right, for drug design, and also the large body of published literatures, right? Where in this area we know that, well, if you happen to study bioinformatics as well, you probably know PubMed as the big literature repository that basically index almost all the major journals in that, cover the entire biomedical area. And if you're just doing data mining and computer science, clearly DBLP is a place to go to find the papers you like to read. And also for other government and private agents, such as in US, the Center for Disease Control, the CDC, and looking at disease transmission, such as influenza and others. And also nowadays a lot of people study social media data, right, and the social networks from the data mining community. But lately, those data are also leveraged to solve biomedical problems. So again, this is one of the traditional problems that many groups have studied. So basically the question is, can we prioritize candidate genes for disease study? Okay, and in my group, we also have developed a lot of different methods, and typically, in particular, we're targeting to study the epistatus effect. So basically, the joint effect of multiple variations, not just a single allele study. Okay, so you can see the computational challenge would be that there's so many potential interactions that one could treat as candidate for the study, right? If you want to design an experiment for each such specific combination, then that's quickly can become intractable, both computationally and experimentally, right? The cost, the time, and the manpower and involved, that's just intractable. And the question is how, if we still want to study that, how do we do it? In our group and together with collaborators, the approach, maybe not the only approach, at least one approach we have been experimenting was that in this pipeline, this is traditional pipeline, we don't want to just follow the and actually unsupervised pipeline, like say, well, you get a lot of data and then you run maybe a typical model is a lasso model or some variation of lasso model to do the association study, okay? And hopefully we can find one or more variant that are highly correlated with the disease outcome. So what we hope to do is to bring in this rich body of knowledge that people have already accumulated over the year and been kept in one of this knowledge base, right? All of them are, most of them are actually publicly available. But the challenge here is that none of the database is complete and noise free, right? They represent actually the accumulation of collective effort from many, many different research groups. And we cannot even guarantee the data there is consistent with itself, right? So, and they're largely incomplete, and then sometimes even biased and noisy, okay? And if you pull data from different databases, and sometimes you may see, well, there's some inconsistency, okay? And so what we want to do is that we want to bring in all those knowledge bases and in a way that we could somehow set as to guide the model such as a lasso or other model that we're using so that we can prioritize what are the candidate genes and the combinations for the study. So that's our goal. So the way we are doing that is to basically set up as a network. Instead of the traditional univariate analysis, so basically you test one SNP or variant at a time and to examine its role and quantify its role towards a target phenotype. What we do is that we build a huge network or graph if you wish. And then each vertex of that graph or network representing one entity. That entity could be a SNP, could be a gene, could be a protein, and it doesn't have to be of homogeneous type. Can be different type. And then the edge between them representing the known relationships, either from observation in the data, okay, or from one of those established knowledge base, okay. And you can see that between a pair of nodes, if there's no edges, doesn't mean that those two are independent, right? It could be just no one have ever run any experiment to examine the relationship between those two, okay? And even if you see an edge there, it doesn't mean they have a direct relationship, like one is definitely regulating the other, controls the other. That could be indirect. Sometimes it's just merely an observation from the data saying that they are not independent, okay? So, and once we have that, we want to set a framework. Actually, we want to learn from these networks. So, I think I'm pretty much mentioned. This is a recap of all the challenges that I've mentioned so far, and the idea is, well, then how do we do this, right? Because the way we construct the network, the way we represent the data seems does not suggest an easy route, right, to find the result we want to find out, right? It's just maybe it look like a different ways of representing things. That we still need to address all of these challenges. So our first work in this is basically say, well, let's not try to be too ambitious, right? Instead, we want to just say, well, let's study either knowledge or data just from two sources, right? As if like if you I want to study myself, like from maybe reconstruct a 3D model of myself, and then but I only have maybe let's look at 2 pictures of me, maybe from different angle, right? To see if we can somehow bring them together, right? To form a little better picture of myself. So if we are able to do that, then we can generalize that to more than 2 domains, right? To more complicated scenario. So here we say that, well, we have the, on the top one, those are the interaction network of the SNPs, the genetic interaction network. And on the bottom is the gene co-expression network of genes, okay? And you can pull out any 2, right, networks, oops, sorry, any 2 networks, and when there is an edge, that means that we have observed some kind of interaction. When there's, and then the edge can be associated with a weight, right, typically numerical weight, and also across domains, you can see there's also some additional edge to indicate maybe one of the SNPs is maybe in the maybe regulatory region, promoter region of the gene, or maybe in the axon or something. There's some relationship there that we know of. And again, we can use an edge to indicate that. And that edge can be of different type, different associated with maybe numerical weight, okay, typically, to indicate the difference between the different type of edges. And then we want to set up the learning. So basically, the idea here is that we want to resolve the inconsistency between these 2 domains, or in other words, to maximize the consistency, right, across them, using graph regularize the learning, okay? So this is a little bit, I mean, I realize this is maybe too early in the morning to show you equations. So, and the idea here is that we have a regularizer, graph regularizer, to control that how much, I mean, basically we want to maximize the consistency or concordance between those 2 domains, okay? And the regularizer just to penalize the inconsistency. So, and then, and of course, and then that's the only part the difference we introduce to inside the lasso model, okay? So as the additional constraint, okay? And then we can examine how well or how badly that performs, right? Does that help? And in this chart, on the top and the bottom, and the top half shows the how well we're doing in identify the cis enrichment, and then the bottom is the trans enrichment between the SNPs and the gene expressions. And our method is called the 3 ones as different variations: the G lasso, GD lasso, GGD lasso. So my student give those names. I guess he ran out of ideas, right? Come up with innovative names. But the idea here, you can see that the number shows the statistical significance of like one method definitely performed better than the other. Okay, and I already sorted this table in decreasing order of the result. Okay, the GGD lasso performed the best, and which is listed on the top. Okay, so this is on using a small yeast data to illustrate, to see that, well, by introducing those constraints, actually it does help, right? The reason we only show the yeast model is many of the some of this other method actually does not scale up to the, a more larger data set. So in order to have a fair comparison, we have to pick a data set that we can get a result from every model. So you can see in this case, the traditional lasso is the worst. So encouraged by this result, we can of course generalize that to multiple networks. Okay, and what I'm showing here is a picture of, actually I just draw 3 domains, and you can see that each domain have can be represented as a graph or network with vertices and edges representing entities and the relationship between them. And again, nodes from different domain doesn't have to be of the same type, right? You could imagine one of them is SNP, the other the genes, and the third one could be proteins. And you can see, I'm not sure you can see it clearly, but there's edges across between every pair of domains, and with weight that. And for the KDD audience, and it's quite typical to use a matrix representation of all of those, right? Within each domain, you can use a matrix to represent the affinity between pairs of entities, and those are the edge weight. And then cross-domain, you have another set of matrix to represent those relationships between every pair of domains. Okay, and in this particular setting, we say, well, if we want to find multi-domain clusters, so basically finding the consistent grouping across different domains, and actually we can use a method developed in our group called, based on the clustering disagreement. So basically we want to minimize the disagreement across different domains. And the idea is that in order to consistently measure the disagreement, we have to map everything to a transferred space, so that in that new space we could directly compare things from different domains, because otherwise it's heterogeneous, right? We cannot compare apples and oranges directly, but now if we ideally just if we want to really compare the taste, then we just maybe grind them and make apple juice and orange juice and drink them and compare them. And so basically, I do not intend you to go into the formula to really understand this. So the idea here is that we have the objective function. Basically, there's two terms. Ideally, one is a within domain term. We want to minimize, so basically minimize the intra-cluster distance, maximize intercluster distance, but also cross-domain. We also have the term to optimize the mapping. So basically, we want the mapping to be the most consistent. Okay, so we apply this to three domains exactly. One is a genetic interaction network of the SNPs, and the middle part actually there's a multiple of gene expression data, right? It doesn't have to be one, okay? Multiple, actually three in this case. And then on the right side is protein-protein interaction network. You can see that's built on the proteins. Okay, you can see these three types of networks. They are of different size, different edge density, right, and different meaning. And then once we bring in this, and then we still want to run clustering compared to other methods, you can see that actually we can identify more significant modules that are highly enriched using gene ontology. The p-value of those discovered modules, and if you look at the curve, the lower to the right, to the corner, the better. And then our method, again, my student gave a name called CGC, performs the best compared to all the alternative method based on the spectral clustering or non-negative matrix factorization. So why we're studying this, okay? And one of the current project we're doing, right, going from there is that we want to be able to study disease, right? We're not just satisfied, okay, let's just study genes and proteins, and then those are nice, and but we want to even go beyond that. So we have a current center, like I think if you're in US, you know the NIH BD2K, so Big Data Knowledge Center. So we want to basically model the disease and the treatment. right, from the symptoms and other things data we collect from the patient. And more importantly, this is what I learned from my collaborator, is that like for us, we talk about a particular type of disease. Let's see, in our case, we study heart disease, right? We think heart disease would be just one disease, right? But in reality, actually, if you talk to any clinician doctor, and then they will tell you actually it's a class of diseases, right? And you can think the disease taxonomy would be like a hierarchy, and then somewhere there's heart disease, and then immediately the next level there's 6 different subtypes of heart disease, and then following that there's a big tree, right, of a more specific disease type, right? So it's not just one disease. As you could imagine, some of the subtypes are very common, right, with a lot of patients. Some of the subtypes are rare, right? We don't have many patients. So now the question is that if we want to improve the diagnosis and the treatment of those rare diseases, what do we do? Can we borrow knowledge from all those common diseases, right? And of course, we counted, it's not that easy to borrow from 2 very different diseases, right? But if they're all heart disease, right, it's just different subtypes, maybe things would be easier. Again, we started by doing something that at least seems to be easy to get started. So in order to do that, we actually build this complex disease network and involve all the levels, right? The very top level would be the disease similarity network, okay? But for each of those, that's all connected to, like on the bottom here, I show you there's different genes and different tissues, because there's always tissue-specific gene networks, right? So it depends on the tissue type. Then it could be different. So you can see that this, again, multiple networks connected together. And then we apply the similar ideas, but a little bit more complicated, and then so that we can use that to model the disease progression. The idea here is that if you know the concept of transfer learning, this has that flavor. So basically, if there's some sub-networks that actually are representing the common mechanism across multiple subtypes, that can be leveraged. So that's the idea. So, and then in this process, we soon realized that a disease is not a snapshot, right? Not actually a picture we're taking, right? A disease is a dynamic process. Many diseases actually last a very long time, like heart disease, right? Many heart disease patients, they suffer from that for 10, 20, 30 years, right? So this is a long process. And then also there's another disease like diabetes, similarly, right? All those chronic disease. So traditionally, when we study this, and in terms of from the data science viewpoint, it's like, well, it depends on how much data we have, and then we can acquire, right? And nowadays, it's more and more likely that we're able to really track the patient. So at UCLA, we have this huge the grant effort really sequencing the patient, but also using clever like devices to really real-time track the patient in hospital and off patient at home, right? So we actually the device there's sensors we built, well, my colleague built, and then so that the patient could wear, and also we use like phones and maybe the Apple Watch and Fitbit, so that to track the patient. Even the prescription drugs we give to the patient, right? If there's a bottle, right, on the cap, there's a sensor. When the patient opens the bottle, and then a message or signal at least is going back to the doctor's office through the patient's phone. Of course, lack of that signal indicating maybe the patient forgot, then the other direction, there may be a reminder sending over. And the watch here, right, can monitor the patient's not only the heartbeat, but also the blood pressure, and also using the a number of attributes or features collected, it can really, I would not say directly measure monitor the heart, but really can almost predict, right? If you, as if you are taking like a EKG of the patient, okay, and then if something indicating the patient is not doing well for heart disease patient, and then you can see that's directly again sent back to the hospital, the doctor's office, and then there will be some really early response from the doctor, not waiting under the patient and going back to the emergency room or going back to the next the doctor visit. Well, originally because the insurance company, well, we were motivated originally was to take this problem was the insurance company stopped paying hospitals for their patient emergency room patient, right? If for the heart disease, if they are they had a heart attack and being sent to the hospital to the ER, and then after some treatment they're being released to home, but if within 60 days from that ER admission, and then the patients for some reason come back to the hospital or the ER again, then that second visit, the insurance company is not going to pay. The hospital have to pick up that cost. So there's huge incentive for at least UCLA hospital to cut down that cost. And then the way to do it is through better, surveillance, monitoring, and intervention, so that we can reduce the readmission rate. Of course, even though our initial target was like, let's try to do well in the first 60 days, right? But then the outcome would be actually, throughout the entire course, the patient is doing better. So that's the goal. And in that process, actually, we collect a lot of data. And you can imagine like time series data, a lot of them. Okay, so once we collect a lot of data, and then we can start to do more things. And one of the ambitious things we want to do is like, well, can we rank to find like what caused what, right? The causality, right? So when this was, ambition of my student, and at the beginning when he talked to me, I said, well, let's be careful not use the word causal analysis, right? Because most of the time we can only detect or measure correlation, right? It's very difficult to basically make sure to verify actually it is causality, it's not just a merely correlation. So, but he want to use that word, and then later I think, well, that's the right word, because in a number of cases that we really demonstrate that we did rank the real cause to be the number one. So as you can guess, is that once we study all this is all time series, and then now it forms like a really evolving dynamic networks, right? Because we observe things over time. And then we, again, want to model the relationship, the observations through networks, right? But this now the networks actually evolves over time. And through this series of observations, we want to be able to identify, A, if there's anything not quite right? And B, in that case, then what's the root cause? Okay, so and then in that, to answer that second question, we typically just give a ranked list based on the probabilities. Yeah. So the idea here is that first, when everything's seemingly right, and we want to first model to capture what are the dependencies, right, between the node in the evolving network. We call them invariant, and then we start to track which invariant actually broke, right? Because basically with the observations already go beyond the expected variance of change. Okay, and once we identify that, because no matter how frequent we collect our data, right, they are still discrete, right? And sometimes there are a lot of missing data because for some reason it's not being transmitted or it's delayed. So it is quite possible when suddenly when we see the next observation, more than one thing's already went wrong. And then in that case, if it's just one thing, that's easy, right? So, but if it's more than one thing, then And then how do we know what is the root cause? In that little cartoon here, that actually that indicate 1, 2, 3, 4, 5 edges, right? 5 links, and then seems all went wrong, and then we want to see which one is the cause. Well, of course, I know a good guess would be the thing in the middle, right? Because it seems that's the only thing in common of all the edges, right? The vertex in the middle, but actually, in many cases, that may not be the case, right? It could be one of the vertex just connecting to one edges, and then that thing just got propagated and trigger other things to go wrong. So actually, in our example, that's one of those cases. Actually, it's not the thing in the middle, and but our idea here is that we want to treat this as we use a matrix factorization, and then we want to sort of like the idea here is that we generate a number of hypotheses for each of the possible causes hypothesis, and then we want to rank those hypotheses or measure the probability of those hypotheses is true based on how well, given that hypothesis, we can reconstruct the observed data over the time. Okay, the one that can allow us to best reconstruct the data, that becomes the top. Okay, of course. That's a very vague idea, but we did all the calculations, and then also calculate the probability and sort the probability. That's the idea. So, and we also tested this not only in the medical domain, but in other domain. Like we test that for the bank system, the power plant system. So, there's a number of very different domains that we all tested, and it works very well. And this one was very recent, published a year ago, right at KDD. So in this exercise, and we also helped the hospital to build a patient system for adaptive retrieval and summarization. The idea here is that through that the early study, we realized that sometimes it's quite difficult to compare patient. Okay. Because each individual patient, the more data we collect, the more we realize that they all look somewhat different, right? No two patients are the same. So, but still, we still want to be able to find what things in common, right? For example, if I see a patient that maybe not, the symptom is not the standard ones, right? That we would observe for, let's see, a type of heart disease. And then I would wonder, if I'm a doctor, I would wonder whether there's such cases in the past, right? Maybe my colleague saw it, or maybe someone from a different hospital have saw it, right? Have seen it. So I want to be able to search that. And but then we say, well, this may not be so easy, but then we want to be able to first be able to model the history, the disease history of each patient, to be able to summarize that. That also means that we need to not only, be able to handle numerical data, all those lab tests or some image or some other like time series data, but also be able to handle text, right? Because those clinical notes there, it's quite messy text because they were never cleaned up. They were never meant for publication. They were originally designed for their private consumption between nurses and the doctors, right? Not being able to share it with the patient. So as a result, there are a lot of typos. There are tons of grammar errors, typically not full sentence. A lot of abbreviations, right? Non-standard abbreviations, right? Because literally they just type it, during the interview with the patient, for example, right? So, and then they didn't have time to clean up. So we want to be able to, but nevertheless, there are things mentioned that maybe correspond to some of the lab tests or some of the symptom of the patient, right? They're not independent in that regard. So we want to be able to link them together. So therefore, we also spend some time in doing natural language processing in a non-traditional way, and then topic modeling, because we want to be able to know what's being mentioned, what are the topics in the patient record, and then LDA was the first thing we thought about. And in case you haven't get a chance to to work in this particular topic, and then this is actually I borrowed this from the original paper of LDA. So the idea here is that if you have not just one document, but maybe a collection of documents, and then you have a budget like how many topics overall you want to have, let's say K topics, but what are those K topics in terms of their distribution, word distribution are unknown, right? You want to learn that. And again, for each document, what is the topic distribution in that document is unknown. That's is another thing that you want to learn through this process. So basically each topic is a distribution of word, and each document is distribution of topics, okay? And imagine that, well, if I could learn like things on the left side, then I can use that to help to for me to summarize my text. So maybe in that patient record, and there's some mentioning about having fevers, or there's some mention about chest pain, there's something like that, right, as a topic. So that's our wishful thinking. And so this is the plate notice of LDA, right? You can see it's a very simple setup. And so this again borrowed from the original paper. So the idea here is we have those learned topics, in this case 4 topics. If each one happen to be naturally, corresponding to a, known concept that in doctor's mind or patient's mind, then we can easily use that. But unfortunately, those things, right, that we need in order for us to do the summarization are not available, right? What is available as outcome of the LDA would be this word distribution, right? And if you look at that, most of the time, at least for me, I don't even know which topic or how do I even pull out a keyword or a couple of them to represent each topic, right? Basically, those topics do not correspond to established concept, medical concept or otherwise, okay? So in order to make that better, right, because otherwise we cannot tell the doctor to say, well, forget about your medical training, use my topic. That doesn't work, right? And then my doctors always want to say, well, find some things that he feels comfortable, right? He can understand and comprehend. So the idea we do is that we don't want to just to say, well, let's do just label, supervised LDA, right? But then because we don't know which topics are there, and the potential, the superset of all topics is huge, right? And literally the UMLS, right, the big ontology set, anything from there could be, right? And then we can't just and also we don't even know how many topics are there, right? Typically, in the LDA setting, supervised and unsupervised, you want to have that input parameter there. So what we do is that we take the UMLS as the potential superset, and then we query Wikipedia. Why we query Wikipedia? Because we want to set a better prior for the word distribution of the topics for the medical concept. And we all use Wikipedia quite extensively, right? Because if we, at least in my research, if there's some term I don't know, right, and then I Google it, typically the top one is a Wikipedia page, and then I read that article, right? It's a very nice, clean explanation for non-expert, right? So we want to be able to use that, but we don't want to completely constrain ourselves from the word distribution of Wikipedia, because it is true that if you ask a different person to write that piece, right, the word they use can be very different, right? So we just want to use it as a prior to give it a little bit edge on that, but we don't want it to be constrained. The model we introduced called source LDA, and it's actually, in terms of this, you can see the right-hand side is the traditional standard LDA. The left-hand side are the new things we introduced. And if you don't need to look at the details, so basically the number of things we considered, for example, each topics, right? So I mentioned that if you ask two people to write an article about the same topic, they may use different word distribution. It turned out to be that difference depend on the topic. For some of the topic, people are using quite consistent word. For some of the topic, actually, it's quite different. Okay, so that's why we even introduce another parameter to see to give this variance to different topics and things like that. Those are small things that we introduce to make sure the final model works well. And the knowledge source here that we use BioPortal for the ontology and Wikipedia together with Wikipedia. Okay, and so this is a small data set we want to show how well we can do if we do this, and compared to other, supervised and unsupervised LDA. And you can see source LDA. This is out of 1 million topic assignment to see if it's correct, and then we can do about 700K, 70% correct. But if you look at the other alternative method, even the supervised LDA, the different version of that, they perform quite poorly at the moment because it's a quite challenging task. We're still working on that. So, and then in this process, we also noticed that since the doctor's notes are not clean, right? And we start to look for a cleaner version to help us to train the model. And one of the cleaner source would be, and there's a class of publications, if you look into the PubMed, it's called the case report. So basically, they are papers published on rare disease cases. They're written typically by doctors, right? If they treat a patient, and this is a non-standard patient, not the textbook version, and then they write it up, right? They describe the patient. If you look at the first paragraph, this is like 56-year-old woman was admitted to a regional hospital for increasing chest and abdominal pain after falling from the stairs, right? And then they start to initial screening, does this, and blah, 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 the finals. So it's a long article, right? So we said, well, maybe we can use this for better learning training to be able to model that. So what we did is that we convert that to a graph to a network, right? Because so far we have been trying to convert everything to network, and then we make network connect to network so that we can do all the inference and learning on top of that. You can see for this, there's a different type. There's of vertices, and then there's a disease, there's a symptom, there's maybe the diagnostic testing, right? And then there's also maybe therapeutic treatment, there's intervention, there's all, and the outcome. So we want to be able to do that. This process, at least there's no grammar error, hopefully no grammar error, but still the challenge here is that if you read this, the sentence tend to be quite long and with many medical terms, and also typically when they describe things, so you can see there's multiple modifiers of the final either symptom of disease. And from I'm not trained in medicine or anything, like when I read this, I talk to my collaborator, I say, well, you have to annotate, tag this. I mean, for me, like, how should I classify these things? It's like, it's quite challenging. So in this process, you can see that we need to recognize the entities, the concept, and then identify the relationship. If they say, well, this happens after that, or this might be the cause of that, things like those. And we want to, and even for example, the way patient describe the pain, right? Because you can see that pain is very common for many diseases, right? But there's so many different types, right? How the patient feel, right? Different severity, different body part, and then sometimes there's patient could say that, well, the pain, right, is better maybe in the morning, but then after you get up and doing some movement, and then it's getting worse and things like that. Or maybe before and after meal, it feels differently. So there's so many important details, right? We can't just say, oh, here's a pain, here's that at that level. We have to precisely describe those in the in the network, and then we doing the typing, and then this would be like, again, in these networks, and in that one is like RAS. Actually, there's ambiguity in this example. That could be either a syndrome or I think a protein, right? But then it's appear in the same sentences. Actually, there's two different meaning of the same abbreviation. So, but then we want to be able to correctly figure that out. So what it is, right? So that we can properly label the graph that for later analysis. And recently, one of my students did this study. So basically, because a lot of time we don't have a lot of good text, right? So that for the training. So he's a Chinese student. So he pulled out both texts in English and the Chinese language, and see that, well, can we set up a deep neural network model to basically train on both? So the first we said, well, we want to do it in multi-domain, right? It doesn't have to be from the same paper or the same source or same doctor's notes, right? Could be can we bring together one paper, one doctor's notes, and some like blog online, things like that, right? So, and then in this case that, well, he's going to go wild and said, well, let's do it even for different language to see if we can do it. It turned out to be actually, once you bring multiple things together, it performs better, slightly better than just a single source. And there are so many different models, mostly deep neural network models proposed. This is a standard benchmark, just to compare the performance. The Omni RCNN is our model, and you can see that at least I can show you the F1 score is better, because we are still continuously improving this. And the reason we pick this is that we don't have to rerun other people's model, because everybody uses the same benchmark. We can just compare the F1 score directly. Just to show you a few examples to see that this is how to identify whether we can correctly identify the relationship in the sentence, the type of relationships in the sentence. And then I just show you the distribution of importance of each word in characterizing that relationship. Okay, the relationship could be something is caused by something, or the cause effect, or the message topic, or entity, destination. There's a different type of relations, right? And then not only we can do it right, and it seems like we also show the trigram that carries the most weight, and there it seems to capture the right part. Okay, so what we do here is that we want to build networks, right? And sometimes if you talk to Google, they say the knowledge graph and the knowledge network. So idea here is that from different data type, we can always build graphs to represent what we observe in the data, what we learn from the data. And even from established knowledge base, right, we can always find a way to map them into a graphs, right? And the beauty of doing so is that it gives us the opportunity to link them together properly. Okay, so once we bring things together, right, match them together into one joint graphs, and then we want to be able to, for example, link symptom sign with with the genetic cause, or with something like molecular signal and things like that. Okay, and then it's a huge, gigantic graph, and then there's a number of things we study, like natural language processing, topic modeling, semantic analysis, to be able to allow us to build better graphs. And what I didn't talk today was that I also collaborate with my colleague to do the image analysis, because there's a interesting source like the medical image plus the report from the radiologist reading. Okay, then that's another source. And also we do a lot of modeling on the networks. And this is my colleague Joe, who's somewhere here in the conference, MetaPath and MetaStructure. She does a lot of those development. And in my group, we're doing a lot of multi-view clustering, network embedding, and then to be able to do the inference on the graphs. So this is an integrated view. Of course, there's multiple data sources, and then you can always add more. And it's not just one loop. It's really iterative process. And we want to be able to construct multi-layer graphs, okay? And to be able to link them, use that to hopefully provide the clinicians and other domain scientists empower them to make a more and better like hypothesis, right? Doing better diagnosis, predictions, surveillance, monitoring. So that's our goal, okay? So you can see that I showed you like, different aspects of this whole picture, right? So to see that how we can, step by step enable this, okay? This is going to be my last chart because I just want to show that actually even that what I show is this is a little bit under the hood what we're doing from 4 layers application from different data type, and also the analytics layer, which is my group is doing a lot to build different analytics model. And also what I didn't get time to talk would be the infrastructure layer and the hardware layer. We also have project. So basically going really vertically across this way, right? So that the question is that how do we design better hardware, right? Customizable hardware and infrastructure to better enable those analytics effort, right? So that's what we're also doing. And then, so this would be my, last acknowledgement. Of course, this won't be possible without my student and collaborators and funding from NSF and NIH. So I'm just the one that sit back and watch everybody doing things, and once in a while I give talk and I hide myself to write proposals. But I have great students and colleagues, so they did most of the work. Thank you.